How many proofs are there in support of the theory of evolution? That is the question. And the answer is simple: millions. Today we'll take a look at one. I promise it's new, it's not a repetition like the sign, line, ALU or ERV insertions that the YouTube is flooded with. Protein kinase is an enzyme which catalyzes the phosphorylation of a downstream target peptide. pKa has two catalytic subunits and two regulatory ones. Being activated by the CAMP, the catalytic subunits phosphorylate the target peptide in the threonine or serine residue. All pKa catalytic subunits have a motif called glycine lead. This motif holds the ATP in a fixed position during catalyzation. The consensus sequence of the glycine lead is GXGXXGXV, where G is glycine, V valine, and X any amino acid. Notice the theory of evolution has a scope to make predictions here. Firstly, all the X residues are free to be any amino acid. As organisms share common ancestors, there should be a tendency of those X residues to be homologous in the closely related organisms and gradually diversified in the distantly related ones. Secondly, even if the closely related organisms share common X residues in their glycine leads, as it is possible for one amino acid to be coded by up to 6 codons, the codons coding those X residues will be homologous in the closely related organisms and gradually diversified in the distantly related ones. How can you confirm these predictions? The first prediction can be confirmed using bioinformatic tool homologene, and the second one using BLAST. Okay, so let's start. We need to go to the NCBI website to do the homologene. So type in www.pubmed.com. Here we are. Choose the homologene database and type in your query term, which is protein kinase A catalytic subunit. Here is the result, and notice the order. It's human, dog, cow, mouse, rat, chicken, zebrafish, wall crease again, and finally rice. So let's take a look at the multiple alignment. Here it is, and the glycine lead should be somewhere here. We need to scan to find out where it is. GRV no, GHY no, GDT no, GVG, TFGKV, yes, we have found it. It's here. These 8 residues are the glycine lead. GXGXXGXV. Notice here the homologene result accurately confirms the prediction we made. We human, along with 2 other eutherian mammals, mouse and rat, 1 amniotic chicken, and another Nafan zebrafish share a common glycine lead, composed of the sequence GVGTFGKV. In the Viridiplantae, we see the first amino acid substitution, as the wall-crease glycine lead contains leucine instead of valine. In the second wall-crease, there is another substitution, where leucine is changed into arginine. The other Viridiplantae rice shares the same glycine lead with the first wall-crease, so the findings exactly fit with the prediction of the theory of evolution. Now let's do the BLAST. Let's go to the gene database. Here is the report. So this one, protein kinase CAMP dependent catalytic beta Homo sapiens. That one sounds nice, so let's take a look. Here it is. We need to scroll down. This is the RefSeq section, which lists all the isoforms of a gene and the respective mRNA and protein products. So I'm choosing the isoform 1, and let's take a look at the protein. Scroll down. Here is the sequence, so the glycine lead should be somewhere here. Let's try to find out where it is. GFA no, GTA no, GTGSFGRV, yes, we have found it, it's just here. So where it starts from? 60, 70, 80, 90, 98. So in the protein chain, the glycine lead starts from the 98th amino acid. Just remember that. Now let's take a look at the mRNA. Scroll down. residues. That's what I'm looking for. Here is the translate, and it's telling me it starts from the 94th nucleotide. So let's scroll down. In the 94th nucleotide, we should find the start codon. So 60, 70, 80, 90, 1, 2, 3, 4. Yes, we have found it. ATG, which codes for methionine. So the translation starts here, and the glycine lead should be somewhere here. We need to calculate to find out where it is. So in the protein chain, we have seen the glycine lead starting from the 98th residue. So 98 minus 1 times 3, 291. And there are 94 nucleotides before the start codon. So let's add 94 nucleotides, 385. So the glycine lead should start from the 385th nucleotide. 360, 70, 80, 1, 2, 3, 4, 5. Yes, GGA. This codon codes for glycine, so we have seen the glycine lead starting from here. Now I'm going to do the blast. I'm going to do the protein blast first. So scroll up, click here. This is the query form. That's the accession. Make sure that the database is correct. So I'm doing a protein blast. It's NR. Yes, correct. Select a target organism. I'm choosing chimpanzee. So let's do the blast. Here is the result. Found quite a lot of target sequences. Let's take a look at the first one. So it's CAMP dependent protein kinase catalytic beta pan-troglodytes, right? And we know that the glycine lead starts from the 98th nucleotide, so it should be somewhere here. Yes, it's here. GTGSFGRV. Notice this query sequence aligns accurately with the target sequence. There is no substitution or anything like that. Now let's do the nucleotide blast. Scroll up, click here, choose the appropriate database. I'm doing a mRNA, so I'm choosing that. Target organism, chimpanzee, and blast. There's the result. Notice here there are quite less results in number than the protein blast. It's because in protein there are 20 options for a single position, because there are 20 residues, but in nucleotide there are only 4. That's why in bioinformatics, when we are doing sequence search, protein is preferable. So let's take a look. Pan-troglodytes, similar to protein kinase, CAMP dependent beta, right? And we know that the glycine lead starts from the 385th nucleotide, so it should be somewhere here, starting with the GGA codon. Here it is, yes. So it should be the glycine lead. That is the glycine lead sequence. And notice all the nucleotides align properly with the target sequence. Now what I'm going to do is I'm going to do blast in 2 other organisms. I'm going to do it in mouse and zebrafish, both protein and mRNA. Then I'll post a screenshot of those, and then we will analyze the data. Now let's analyze the data. All glycine-lead residues are identical in human, chimpanzee, mouse, and zebrafish. However, the codons coding those residues vary. Human and chimpanzee share all the identical codons. Mouse and human share 5 identical codons out of 8. Human and zebrafish share only 1 identical codon. So the pattern predicted by the theory of evolution is evident here. We have confirmed our prediction.